Today we are going to learn dynamic light scattering and zeta potential analysis. In today's lecture, we'll be learning about what is dynamic light scattering and zeta potential, and we'll be also learning about what are the applications of DLS analysis and what is the importance of zeta potential. At the end of this lecture, by practical demonstration, we'll be learning the DLS and zeta potential analysis. Let us see what is dynamic light scattering. Dynamic light scattering, also called as photon correlation spectroscopy, and this is a technique to measure the size of particles in suspension or solution. Here the size are determined by analyzing the fluctuation in the intensity of scattered light caused by Brownian motion of the particles. By using DLS, we can measure the hydrodynamic radius and size distribution of the particles, and also we can measure the diffusion coefficient. Let us see the principle involved in DLS and how we can use DLS to measure the particle size. The first one is the Brownian motion. Brownian motion is the random movement of particles in the suspension due to their collision with the solvent and other molecules. Here the smaller particles diffuse faster while the larger particles diffuse slowly. And next one is Rayleigh scattering. While the particles are in Brownian motion, they scatter the light when illuminated by a laser beam, and the scattering happens as the size of particles is smaller than the wavelength of incident laser beam. The intensity of the scattered light fluctuates over time due to random motion of the particles, and the correlation function in the instrument that analyzes the decay of fluctuation in the light intensity over time and provides the information about the particle size. Let us learn about the various parts of DLS and its function. In the DLS, we have the laser source, which provides the monochromatic light, followed by we have the attenuator, which control the intensity of the laser beam reaching the detector. And here the sample cell where we have to keep our cuvette containing the samples. And we also have two detectors, and these detectors are photodiode detector. It is used to measure the scattered light, and it is usually placed at 90 degree and 173 degree. Then we have the correlator. So the correlator analyzes the fluctuation in the intensity of the scattered light signal. Then all the data can be analyzed with the help of the software in the computer. Let us learn about the function of correlator in the DLS. The correlator is the computational brain of the DLS system, which is basically a signal comparator, and that measures the fluctuation in the intensity of the scattered light at a point over a period of time, and then correlates it to determine the diffusion coefficient of the particles. And this diffusion coefficient of particle is directly linked to their size through the Stokes-Einstein equation. The larger particles will have the slower fluctuation. As you know, the bigger size particles, it moves very slowly. So the larger particles will have the slower diffusion and longer correlation, whereas the smaller particles will have the rapid fluctuation, that means rapid diffusion and rapid correlation. The Stokes-Einstein equation is the theoretical basis for DLS measurements for interpreting the particle size. If you want to measure the hydrodynamic radius, we have to use this equation, and the hydrodynamic diameter can be analyzed by using this equation. Let us see what is hydrodynamic size in detail. The hydrodynamic size represents the diameter of a hypothetical sphere that diffuses at the same rate as the particle being measured. That is why this hydrodynamic size is not the actual size of the particle, because it includes core particle size along with the molecules adsorbed on the particle surface. It may be surfactants or polymers or ligands, and also solvation layer, that is the layer of solvent molecules adhering to the particle due to intermolecular interaction. The hydrodynamic size is also affected by the solvent viscosity and solvation effects. So that is why whenever we measure the size using this DLS, that is we are measuring the hydrodynamic size, which is slightly bigger than the size which we are getting from the transmission electron microscope. The size which we are getting from the transmission electron microscope is the actual particle size. For example, if the particle size is roughly around 10 nanometer, and in the hydrodynamic we will be getting around roughly around 20 or 25 nanometer, because we will be also having the molecules which is adsorbed on the particle surface. It may be a surfactants or it may be the some of the ligands which is attached to the particular particle. Let us see what is polydispersity index. The polydispersity index PDI is a dimensionless number that indicates the particle size distribution in a sample, and it provides insight into the distribution of uniformly sized particles within a dispersion. This PDI can be calculated based on the width of the particle size distribution relative to the mean particle size. By using this equation, we can calculate the PDI, where the sigma is the width of the size distribution peak and d is the mean particle size. From this picture, you can understand there are two peaks in this picture. This is due to the size difference in the nanoparticle. For example, the first peak is due to the size of nanoparticle in the range of 10 nanometer, and the next peak is due to the nanoparticle in the range of 100 nanometer. If you are having a nanoparticle of different size, and if the size distribution is very high, so due to which your PDI, that is polydispersity index, will be also high. Let us see what is PDI values. When we synthesize nanoparticles, if you are getting a uniform size nanoparticles, the PDI will be low. It will be between 0.0 to 0.1. And these uniform size nanoparticles are mainly useful for drug delivery applications. And if you are having a PDI between the range of 0.1 to 0.4, that means these are moderately polydispersed system. That means your nanoparticle is having slight variation in the size. And if you are having a high PDI, that means above 0.4, that means the nanoparticle is aggregated, or it may be contaminated with some other particles, or it may be due to the pore synthesis. The particle size is bigger, and also you are getting a broad range of the sizes. Let us see what are the various factors influencing the PDI. The first one is aggregation. When the particles clump together, that leads to increase in the PDI values. And next one is sample purity. If there is a contamination or any dust, that will also leads to higher PDI value. And third one is synthesis quality. If the synthesis protocol is not well optimized, that leads to broad size distribution. And fourth one is measurement condition. If there is a variation in the conditions like temperature or inconsistent sampling, so that leads to different size, and that will affect the PDI. Here the low PDI means it indicates the high quality uniform size nanoparticles. Let us see some of the important applications of DLS analysis. It will be very useful for nanoparticle characterization for understanding the size distribution. In biotechnology, it will be useful for understanding the protein aggregation and biomolecular interaction. In pharmaceuticals, it will be useful for understanding the liposome based drug delivery system. And in the environmental science, it will be useful for analyzing the colloids in the water. In material science, it will be useful for studying the polymer dispersion and emulsion. In the field of cosmetics, it will be useful for optimizing the pigment dyes and various emulsions. Let us see some of the advantages and limitations of DLS. The advantages are, it is a non-destructive and rapid technique and requires minimal sample preparation. Whereas the limitations are, it does not provide the actual size, it provides the hydrodynamic size, and it does not provide any information about the shape of the nanoparticle, and it assumes all the particles are spherical in nature. Let us see some of the common issues in the DLS analysis and how to overcome that. The first one is high PDI. If you are having a high PDI, it indicates aggregation of nanoparticles. So, you can try by diluting the sample. The next one is multiple peaks. If you are getting a multiple peaks, it may be due to the contaminants. You can filter the sample with 0.22 micron filter, and if you are getting a low signal, it may be due to the insufficient particle concentration. You can increase the particle count. And the fourth one is temperature fluctuation. If there is a temperature fluctuation, you can use a temperature control setup. By using this, you can overcome the common issues in the DLS analysis. I hope you got an overall idea about the DLS analysis. Now we move on to zeta potential analysis. Zeta potential is the electrostatic potential at the boundary between the stationary layer of fluid attached to a particle and the surrounding liquid in which the particle is suspended. Zeta potential values determines whether the particles in suspension will remain dispersed or aggregate. If we have high zeta potential, the stability of nanoparticle will also be high. And the zeta potential is not a property of the particle, but of the system, including the particle and the solvent. That is why if there is any changes to the medium, if there is a changes in the concentration of ions, or if there is a changes in the pH, that will affect the zeta potential. Let us learn about zeta potential more in detail. This is the particle and this is a particle surface. Followed by that we have the Stern layer, and the Stern layer is composed of ions which tightly bound to the particle, and these ions travel with the particle. And followed by that we have the slipping plane, and this is the radius within which the counter ions move with the particle. And ions beyond this point do not travel with the particle, and the potential at this point is called as zeta potential. Followed by that we have the diffuse layer. The ionic concentration in this layer is not same as in the bulk, and outward concentration gradient of ions exists until it equilibrates with the bulk. Let us see the principle of zeta potential measurement. The main principle behind the zeta potential measurement is electrophoresis, which is the migration of molecules and particles due to the presence of an electric field. To measure zeta potential, a technique called electrophoretic light scattering is utilized. The working principle is the sample is illuminated by a laser beam, and when electric field is applied, the charged particles move towards the oppositely charged pole and attain a certain velocity. The movement of particles cause a Doppler shift in the frequency of the light scattered by the particles. You can see here, if you have a negative charge particles, these particles move towards the positive charge. When the particles are moving, that will cause the Doppler shift, and that will lead to the difference in the frequency of the light scattered by the particles. The changes in frequency from the Doppler shift is used to calculate the velocity of the particles using this equation. Then electrophoretic mobility is calculated, which is related to the velocity of the particles, can be done by using this equation. Finally, by using this equation, we can calculate the zeta potential, which relates the electrophoretic mobility to zeta potential. Let us learn about zeta potential and particle stability. The magnitude of zeta potential gives an indication of the stability of the colloidal system. If all the particles have large zeta potential, they will repel each other, leading to dispersion stability. It means if we have a particles with uniform charge, and if we have the high charge, then these particles repel each other. That means you have the large zeta potential. If the particles have low zeta potential values, then the repulsion force disappear and particles start aggregating, leading to dispersion instability. So, if you have a low charge, the particles will try to come closer and aggregate, and the dividing line between the stable and unstable aqueous dispersion is generally taken at plus 30 or minus 30 millivolt. From this table you can understand, if you have a particle with zeta potential between 0 to plus or minus 10, it means it is highly unstable. If you have plus or minus 10 to 20, this is limited stability. If it is between plus or minus 20 to 30, it is moderately stable, and if it is more than plus or minus 30, this is highly stable. Let us see the role of pH in zeta potential. The pH of the suspension plays an important role in the stability of the particles. Changes in the pH influence the surface charge, ionization of the functional groups, and most of you know this isoelectric point. The isoelectric point is the pH where zeta potential equals 0. If you are having a low pH, that means positive charge dominate on the surface, you get positive zeta potential, and if you are using high pH, negative charge dominates on the surface, you will get the negative zeta potential. At isoelectric point, you have the 0 charge. So, due to 0 charge, you get the 0 zeta potential that leads to particle aggregation. Let us see the importance of zeta potential. The zeta potential is useful in determining the stability of particles in a colloidal system, and in drug formulation, it helps in optimizing the emulsion parameters for better drug stability. It can be also used to study the surface functionalization by analyzing the changes in the zeta potential. For example, you are having a nanoparticle, and if you are using PEG, that is polyethylene glycol, as a capping agent, it will give negative charge, and if you are using a PEI, that is polyethylene amine, if we are using that, it gives positive charge to the nanoparticle. So, by using this zeta potential analysis, we can also understand the surface functionalization, and the effect of pH on the dispersion stability can be also studied by using this zeta potential. And the zeta potential not only useful for nanotechnology and nanomaterials, it also helps in the manufacturing process of paints, pigments and cosmetics. Let us see some of the common issues in the zeta potential analysis and how to overcome that. The first one is poor sample preparation. If the sample containing impurities or dust, that can be removed by filtering the samples using the suitable syringe filters, and make sure that the sample is homogeneously dispersed. You can use ultrasonication if required, and ensure proper dilution concentration within the instrument range. And it may be also due to the incorrect pH or ionic strength. If there is an incorrect pH or ionic strength, that can shift the zeta potential unpredictably. So, adjust the pH carefully using calibrated buffers or acids and bases. The third one is electrode and cuvette contamination, which can lead to unreliable results. So, it is always better to wash the cuvettes with the distilled water, or you can use a fresh disposable cuvette. The fourth one is aggregation of particles, which can alter the zeta potential values. For that, stabilize the sample with appropriate surfactants and avoid sample preparation conditions that approach the isoelectric point. So, as I told earlier, if the sample is at isoelectric point, then it tends to aggregate. The fifth one is temperature fluctuation, which can change the particle mobility and medium viscosity, which alter the zeta potential. So, it is always better to maintain a constant temperature during the measurements, and also record and report the measurement temperature for consistency. I hope you understood the basics and principle of DLS and zeta potential analysis. Let us go to the lab and learn this technique more in detail. In this experiment, we will learn how to perform zeta potential and hydrodynamic size measurements of nanomaterials. In today's experiment, we will measure the zeta potential and hydrodynamic size of tannic acid capped gold nanoparticles, which we have synthesized earlier in one of the previous lab demonstration. For proceeding with the experiment, the materials required are Zetasizer cuvette, 4-sided DLS cuvette, microtips, micropipettes, ultra pure water, tissue paper, wash bottle with ultra pure water in it. First, we will measure the zeta potential of the synthesized gold nanoparticles. Let us start by taking the Zetasizer cuvette and wash it thoroughly. Dry the cuvette using tissue paper. Next, carefully add the sample into the cuvette, ensuring the liquid fills up to the copper electrodes. The sample must touch both electrodes. Now turn on the machine. It will warm up and you wait until the indicator light turns green. Then open the Zetasizer software on the system. Before starting the experiment, adjust the settings and set the parameters. Choose the measurement type as zeta potential. Enter the sample name. Select the cuvette material as polystyrene latex. For the dispersant, we have used ultra pure water. So here we have to choose water. In the general options, select the appropriate model. In the temperature settings, set the equilibrium time to 120 seconds. Choose the cell code corresponding to the cuvette. In the measurement settings, select automatic so that the machine will conduct the runs automatically based upon the type of sample. Click OK in the data processing section. Once all settings are configured, place the cuvette inside the machine. Open the lid. Insert the cuvette and close the lid. Press start. The machine will equilibrate for 120 seconds. Then the machine will optimize before beginning to measure the sample. The machine will take triplicate measurements with the number of runs per measurement varying depending on the sample. This process will be handled automatically by the machine as we have selected the automatic measurement setting. Here you can see the data of 3 measurements. We have to choose all together and press the zeta potential. You will see the data from the 3 measurements displayed, each shown in a different color. The zeta potential of the tannic acid cap gold nanoparticle is 20.1 millivolt with a standard deviation of 5.01. Then finally save the data. Now remove the cuvette from the machine and wash it with ultra pure water. Next, we will proceed with the hydrodynamic size measurement using dynamic light scattering, that is DLS. Open the same software. This time, select size as a measurement type. Enter the sample name. The remaining parameters are the same except for the cell. Since we are using a different cuvette, make sure to select the correct one in the software. Once the parameters are set, add the sample to the 4-sided cuvette and place it in the machine. Press start in the software to begin the experiment. Similar to the zeta potential measurement, the machine will equilibrate for 120 seconds, then optimize before starting the measurements. The machine will take triplicate measurements, and the number of runs per measurement will vary based on the sample. As I told earlier, the machine will conduct the measurements automatically based on the type of the sample. Once the measurements are complete, you will see the data from the 3 measurements displayed. Choose the data together and press intensity to view the graph. The average particle size of the tannic acid cap gold nanoparticle is approximately 110.4 nanometer. Finally, save the data from the DLS size measurement. Save the data. So in this experiment, we have learned how to perform zeta potential and hydrodynamic size measurements of tannic acid cap gold nanoparticles. As a summary, in today's lecture, we learnt about DLS and zeta potential analysis and parts of the instrument and principle. Also, we learnt how to perform DLS and zeta potential measurements through practical demonstration. Thank you for your kind attention. I will see you in another interesting lecture.